Hey everyone, now we're going to talk about transmission of pathogens. And so this is where the pathogen gets onto or into your body so that they can then cause a disease. And there's a correlation between how a microbe is transmitted and the degree of virulence that the microbe will have. So what is virulence? There we go. It's the degree or intensity of pathogenicity of the microbe. So this is determined by how likely the microbe is able to and how long the microbe is able to survive outside of a host. So let's look at different mechanisms of transmission. So there's direct contact. This is typically going to be less virulent. If you have to physically move from one individual to another individual, if you're physically going from one human to another human to cause disease, then you can't cause a disease that is super lethal because the first individual, so individual here, if it was lethal, he wouldn't be able to get to the next individual. Vector-borne diseases are going to be much more likely to be severely virulent and more lethal. And the reason for this, if you have a vector, that vector, so example, again, here's a mosquito right there. If he bites an individual, that individual can get a disease, and let's just say West Nile, right? West Nile encephalitis. So now this individual's in a hospital or in his bed, depending on where you're living, right? And he may die, and he may not transmit the disease. Well, he wouldn't transmit the disease, but that same mosquito could transmit it to another individual and another individual and another individual. So this is much more highly virulent than a common cold. The cold is going to be direct contact. Vector-borne contact is more virulent. So what is an infectious dose? An infectious dose is the amount of pathogen that can infect 50% of a population, some experimental population that we're talking about, in a specific time. And this is going to vary from one organism to another organism. An example is Salmonella and Shigella, which we'll see on the next slide. Hand washing reduces the number of pathogens and so will decrease the likelihood that you will hit this ID50 number. So if you have a smaller infectious dose, then you're going to be more likely to cause disease in that population. Whereas if you have a higher infectious dose, then you're going to be less likely to cause disease. So here we have salmonellosis and shigellosis. Both of these cause dysentery. So gut cramping and diarrhea. Severe diarrhea, okay? Salmonellosis, you need close to 1 million cells to cause disease, whereas shigellosis, you only need tens of cells, so up to maybe 100 cells. So shigellosis has a much smaller infectious dose. Which means it is more virulent. Okay. So I mentioned before that when you get an infection, it can be localized or systemic. Here's an example of an infection that is localized. So a localized infection is going to be found in one area of the body typically. So a boil is an example of a localized infection. Whereas here we have an individual with the measles. The measles is a systemic infection. And so systemic means it spreads throughout the body. You can have diseases that release toxins into the blood. This is called toxemia. And so I just use different pictures in blood to show this. Oh, I'm sorry. Viremia, again, just look it up here and you can see different things in the blood that can cause viremia. Or septicemia, where you have multiple agents that is in the blood. Okay, so if it's a virus in the blood, if it is bacteria releasing toxins in the blood, or if you have multiple agents or agents that are going to be life-threatening, that would cause septicemia. So in the 1700s, we started learning that there were bacteria, and we learned, hey, bacteria likely are causers of disease. And so towards the end of the 1700s. But it wasn't until the mid 1800s, about 1860s, when Cook's postulates were produced, and he helped us to identify specific causative agents with an infectious disease. And we've went through Cook's postulates in the past, so I'll go through them really fast again. Microbes have to be present in every case of the disease, and they can't be in healthy hosts. Microbes must be grown in a pure culture. Microbes, if we infect a healthy individual with that potential pathogen, that organism must be susceptible and get sick, and we must be able to find that same pathogen in the newly deceased organism or newly infected organism. So this makes sense. And it's how, or something we use today to still help us identify diseases. But there are limitations. So we know that there can be healthy carriers. Typhoid is an example. Mary Mallon, right? She was a healthy carrier. So we know that you can't always have every single organism that is infected get sick. We also have problems with growing microbes. Some organisms can't be grown on laboratory media. An example would be syphilis. So Treponema pallidum, which causes syphilis, can't be grown on normal media. Some diseases are caused by multiple microbes together. So periodontal disease is an example of a disease that is caused by multiple organisms. So you can't say, oh, well, periodontal disease is caused by this, because it's not. And we can't use humans to grow certain microbes, and you can't find animal hosts always for testing, and you can't use humans. That would be unethical. So you can't run these postulates on all organisms. So we use molecular Cook's postulates, in which we're using molecular techniques to study instead of the microbe, but the microbe's virulence factors. And so in this, we look at the virulence factor gene that's found within pathogenic strains. We then mutate the gene to disrupt the function, and when we do this, we assume there's going to be a reduced virulence. If there is, that's good. Then we can do a reversion or replacement of the gene, and that restores the virulence. Then we know if this specific gene here, this specific gene here is what is causing this disease over here. I am going to stop on this video and we'll get into adherence, so attaching and colonization in our next video. Bye.